This conference will now be recorded.
A very good morning, good afternoon to everyone present here for virtual today. Are you able to hear my voice? Am I clear? Hello, this is David Austin here.
Dear Dr. David Austin, I have your voice.
Dr. Falik. Falik Waiva, are you able to hear my voice?
Yes, I'm here.
All right, all right. Just want to check whether you guys are able to hear or not. All right, all right. So let's start. So, a very good morning, good afternoon to everyone present here for virtual today. It is very great pleasure that this is David Austin, a event organizer. Welcome you all to the virtual meeting of our conference, which is International Conference on Plant Science and Molecular Biology, and also the Agriculture and Horticulture. Well, while we may not be physically together, technology has given us opportunity to connect, share, and grow across borders and time zones. With this meeting, we will engage in thought provoking discussions, insightful keynote sessions, and valuable networking opportunities. We are honored to have an exceptional lineup of speakers and experts who will guide us through emerging trends, challenges, and opportunities. I encourage each of you to actively participate, ask questions, and exchange ideas because the strength of this conference lies in the collective wisdom of its attendees. Before going with today's sessions, I would like to take the opportunity to give the brief introduction about our organization and instruction for the meeting. C2P Forum, our organization name, and Connect to People is the forum where diverse and collective ideas will meet to shape future research. Our platform intends to stand as viaduct between research and researchers. Our aim is to grow network of peers, including students, researchers, and research experts, by exchange of ideas and insights at one meeting point. Our conferences will be going to be held at various locations in Europe, Asia, Middle East, USA. Our purpose is to be the worldwide leader in advancing the field of medical, clinical, engineering, and technology through collaborative research efforts. Through incredible diversity of experiences amongst our board members, CTP Forum facilitate interaction, knowledge sharing, and exchange in global stage through valuable and inspirational connections by facilitating networking. Now, as of our attendees are new to GoToMeetings app, so I will explain you the important settings and procedure how to begin with that. As you have joined through the link provided, you can see the screen where you can find four icons at the bottom of center of the screen. Now, Mike, click on that to mute or unmute yourself. So I request all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. Camera, you can use the button to share or unshare the webcam. If you have the option to share the webcam, you can do it, or else it is completely your choice. The next screen, you can see this button to share your screen. At your schedule, when I will call your name from my side, I'll be making you the presenter. When I'll give you the access to share the screen, then you need to click on this button and you will find the pop-up window which will ask you to share the screen, and you can proceed further. For MacBook and Apple device users, once I make the presenter, then you need to give the permission to share the screen in your system preferences. After that, you need to log out from the session and need to join again from the same link and share the screen for presentation. The next option is leave. This is your last icon. Clicking on this button will leave you from the session. On the top right corner, you can find three icons: people, chat, settings. People, which shows you all the attendees of the webinar. The next, chat. In this option, you can chat with any other participants and with me for any queries. The next, settings. You can see this button to check for settings for a speaker and microphone. So as I mentioned, kindly remain muted during the presentation so that the presenters don't get disturbed. And for any questions, you can ask me or you can write in the message box too. Finally, I would like to request all to give a brief introduction about yourself before starting with session so that we can know about you virtually. Now let move into our session. The first presenter is Tawfiq Shawal Yusuf. Are you ready?
Yes, I'm
All
ready.
right. All right. Mr.
Can
Tawfiq,
everyone hear me? Yes.
yes, we can hear you. And
All right.
rest of the participants can be muted. And please listen to the Tawfiq's your presentation. Mr. Tawfiq, please introduce yourself and you can start the presentation.
All right. Thank you very much for this platform. I wish to state that I'm very grateful for the opportunity given to me because of some financial issues. And I thank the conference organizers for getting it up to me to be part of this conference. Well, my name is Taufik Swali Yusuf. I'm from Ghana, but currently studying in Portugal. My master's, in the last end of my master's. Before moving to Portugal, I worked as a research assistant in the Kuku Research Institute of Ghana. And currently, I am here, been 2.5 years in Portugal. I am a research assistant in the molecular biology lab of my school, Polytechnic Institute of Bragança. Basically, that's what I can say about myself. Thank you. And now I think I am ready to share my presentation. And this presentation was actually an extract from my master research thesis, and I'm very grateful to share it here. And this presentation is going to be my first oral presentation in a conference, and actually my second conference in my research career. So this is very important to me. Thank you. At this moment, I would ask that you share with me, you give me access to share uh, my slides, and then I should be able to start the presentation. David. Hello.
Yes, Dr. Tawfiq, you can start your
Yes.
presentation. Yes, we can hear you.
All right. I don't have access to share the presentation. I don't know.
I'm sorry, just a moment.
All right.
Yes, now you can share the screen.
Is everyone able to see my screen?
Yes,
Okay. Okay. So today I will be presenting on a reliable molecular diagnostic tool for CA90. That's CA90. It's hybrid identification through microsatellites. Yeah. On this, in the left-hand side of my slide, you can see the name of my school, Polytechnic Institute of Bragança, Portugal, and with an affiliation to CIMO, this research center, that's a mountain research center, one of the best research centers in Portugal. Yes. Today I will quickly run my presentation, and it's going to be like introduction, materials and methods, results, and maybe some questions. Yes, just an overview of an idea of what I'm trying to present. Chestnut, for some of us who may not know what chestnut is, on the left hand side on the above picture is just some young chestnut plants that are growing healthy, and in the middle you can see an entire big tree that is diseased, as you can see, because it's not functioning very well. And on the other side, on the left side, you can also see the back of a chestnut with some kind of orange structures on it. That these orange structures are fungi that affects this chestnut bark. And then on my right hand side, you can see some images of the chestnut, the nut itself. That's the edible nuts. And so that was just a quick overview of what the chestnut tree it looks like. Yes. So, for the introduction, I would talk about chestnuts being economic trees that are cultivated by seedlings or vegetative propagation. And they are best known for their valuable timber, nuts, and forest biodiversity. It's a diploid with 12 chromosomes and basically classified under the family Fagaceae with Quercus and beeches in the same family. They belong to the genus Castagna, and today we will be talking about the genus Castagna, but particularly we are going to talk about the Icastanium. We also have the Hypocastanium and the Castanum. But I think that the Eucastanium is the, the question of importance today. And so, chestnut is cultivated mostly worldwide in America, Asia, and Europe. And then the Eucastonium includes species that holds greater economic significance. That's why we are here to talk about it today. And the species are named or belong to certain regions of the continents of the country. In Europe, the major one is the Castagna sativa, and in America we have Castagna dentata, Castagna mollissima in China, and Japanese crenata. And among these species of chestnut, uh, the Castagna sativa and the Castagna dentata in the 19th century was actually distracted by these fungi and some omysets. But however, the Japanese chestnut and the, the Chinese chestnut were a little bit resistant to these diseases. And so I would like to talk a little bit about chestnut production. As you can see on my left-hand side, we have countries that produces chestnuts, with China being the most, producing about 1 million to close to 1,600,000 tons per year, followed by Spain, and in that order. And then on the right-hand side, you can see the global chestnut market statistics. And the current growth rate of chestnut is at 3.1, estimated by IMAC. In 2014, it was estimated that the production was at 3.8 billion, and we are expecting to have a rise, as you can see in the graph. It's expected to rise from 2025 until 2033. We should be hitting 5 billion US dollars, and this indicates how important the chestnut is, and therefore they must be conserved. However, they are susceptible to invasive pests and diseases. Like I said before, at the end of the 19th century, these trees were hard hit by these omysets and fungi, and it was estimated to have killed 4 billion American and European chestnut trees. The ink disease, known as the ink disease, affects the roots of the plants, and the pathogen involved in this is an omyset. known as Phytophthora cinnamomi, and the chestnut blight, Grifonectaria parasitica. And so in that image, with the small root, you can see that the root is black and with some ink coloration, that the root has been affected by Phytophthora cinnamomi, and you can see the effect in the other two pictures. So the plant eventually dies if nothing is done about this situation. And on the right hand side, you can see that orange pictorial stuff. That's Grifonectaria parasitica. And what Grifonectaria parasitica does to the plant is that it attacks the bark and then it eats up the cambium and preventing water and other nutrients from crossing the area that it affected. And therefore, the plants die up, and then you see that the down part of the plant sometimes still exists, but the upper part does not have access to nutrients, and thereby they are not able to survive. The effects of the pathogen and other factors on the chestnut production. You can see in this graph on my left hand side that from the 1960s, the chestnuts were performing very well until the introduction of these chestnut pathogens. That's the Grifonectaria parasitica and the Phytophthora cinnamomi, led to the reduction in production. Then you can see that there was a sharp decline until 2015, where we have a raise in production again, which was due to certain factors. In fact, the FAO actually estimated that the chestnut was chastised in a decline by 2,554 tons from 1961 to 2025. There were other factors that also led to the decline of this chestnut. That was a crop replacement, as potatoes and cereals, because some farmers were not able to deal with the effect of those pathogens. There was also rural population decline, and Asian chestnut groups also suffered from abandonment, natural dieback, and other pest and diseases. And now from 2015, the increase was attributed to influence of climate change and some kind of research and breeding efforts that were put into these plants. So the introduction of these pathogens led to an establishment of hybrid chestnut plants uh, that were resistant clones to this pathogen attack. And so in Europe, there was a strategy about the breeding programs that were initiated in Portugal, France, and Spain to establish hybrid tolerant plants to tolerant to ink diseases. Hybrid chestnut from Asia. So it was a crossbreed from crosses from Asia and Europe breeds. That was sativa and crenata crosses, and they were proven to be notable resistance to the ink disease. Well, some of the hybrids you can see here that I mentioned in my presentation. And what is the problem statement and the objective of my research? Um, the chestnut hybrid identifications are currently based on morphological features and field observations by farmers. Roostocks, and you can see that down here is the prices of the hybrids. They command high prices, and the non-hybrids are cheaper because the non-hybrids are susceptible to the pathogen attack. And so what happened? Rootstock vendors sometimes offers Castagna sativa, which is highly susceptible to the plant pathogen, and ascertaining that they are hybrid cultivars, leading to pathogen attack over the years. So objectively, for my master studies, I decided to take this upon myself and to try to study and to develop and validate a genetic identification tool using microsatellite markers to distinguish ink disease resistant chestnut hybrids from other varieties to enhance the selection and development of resilient rootstocks. So one of my objectives was to select the most effective SSR primate combinations to differentiate the Castanha cyanoventi, which is one of the important hybrid in Portugal that all the farmers currently farm. And however, they are not able to distinguish it, easily distinguish it from the Castanha sativa. And so sometimes they get from the rootstock sellers the sativa, and whilst they think that is cyanovet, and they when the years go by, the plant get attacked by the pathogen and disrupt their farms. There have been, by my research, there have been some molecular identifications of chestnut since in the 90s, and the first one was by isozymes, the followed by random amplified polymorphism, ISSR, and single nucleotide polymorphisms, express sequence tags, and recently, in the last 20 years, microsatellite markers have been used for accurate estimation of genetic diversity of the populations. And so here we come to the materials and methods. What did I do? I researched in literature and found some important papers. And what I did was that from the papers of Santos, which was one of the good papers in chestnut production, they already had some SSR markets, 43 of them, and then I selected nine primates and modified them from these study. A total of 35, I sampled a total of 35 representative samples of hybrids and traditional Portuguese varieties from different parts of Portugal. And then you can see in the last part, Dayfield Green Biotechnology is a commercial breeder who sells these rootstocks, so that I could have a variety of different materials. What was my selection criteria? I selected primates with null allelic features and low observed heterozygosity values from this paper, ranging from 0 to 0.5, because the goal was to target specific genotypes in disease-resistant hybrids. And the primer modification, I had to modify the primers, and the modification was based on the addition and subtraction of one or two nucleotides from the 3' end or the 5' end of each primer to ensure specific binding. Then the methodology was simple. I extracted leaves. I extracted from chestnut leaves the DNA, PCR amplifications. I did gel electrophoresis, and then I sent for Senja sequencing. And from then I did some bioinformatic analysis using our programming, MISA Web Genetic for MISA Web for genetic identification and SSR motifs, and I also use Mega 11 for clustering specificity in order to be able to select from these nine primates that I used. You can see the primates here. The best pair of primates that are able to identify these crops. All right, so the bioinformatic tools that I used here was BioEdit. That's when, right after sequencing, I used BioEdit to visualize and edit the biological sequences. NCBI was used to blast and compare to the sequence, my sequences to the database. And then I accepted above 95% as the threshold, 97% as the threshold. And Mega 11 was used for sequence alignments, and MISA was used for the detecting of SSR motifs, results, and discussion. And so here in my left hand side on this heat map, you can see that in the red color, when you see red, it means the presence of a band, because the primates were basically just for non-allelic features, like to show if there were presence or absence of bands. And so A, B, C was my control, whose genetic situation, whose genetic structure was known. There were samples that we knew their genetic makeup, and so they were used as controls. And 1, 2, 3 until 9 was from a different farm, and then the rest was also from another farm, and the rest from Dayfield Green biotechnology. And so if you look at the sample ABC, you can see that, and then on the left hand on this side of the graph, you can see the primer locus, where I named my primates CP1 to CP11, and you can see that ABC had very good amplifications in all the primates, because you can see the whole of this territory black. Wherever you see is black, it means that there was a band present. However, in sample D, which is known as sativa, which is highly susceptible to these pathogen, these homicides, Phytophthora cinnamomum, you can see that in CP4, the primer CP4, there have not been any band. There was no visible band in sample D, which is sativa, and but in the rest of the other plants, in the rest of the other plants, you could see that there were presence of buds, and this proves that those plants were not they were not sativa but cyanovent. So on my left hand side, you can see that I try to do a phylogenetic tree, depending on the clustering, how well these primates cluster, and to be able to help me to identify and choose some primates to be able to identify or differentiate the cyanoventi from other chestnut hybrids based on their clustering specificity, so that even though I can see that we can see in the heat map that all the primates have good amplifications in the cyanoventi sample ABC, but however, to be able to identify them for cost and time management, we need to have selected some few of these primates to enable us to do the identification as fast as we can. And so, what happened to after I submitted all these sequences to the various databases, trying to figure out what results I could get. The band behaviors that I see was CP2 until CP11. All of them get good band amplifications in cyanovent control. CP4 bands showed bands exclusively in cyanovent, and CP they had the same SSR motifs, even though some of them had variations in the motifs. And so the genetic variations, I observed a total of 18 alleles for the 9 losses, and 27 amplified bands were sequenced, where we identified 31 SSR cells across 22 SSR containing sequences. Trinucleotides were like 67.74% repeats, and then 22.5.8 were dinucleotides, and mononucleotides were 6%, and the hexanucleotides were 3.2%. Here, the marker CP2 and CP2 had the lowest number of alleles. CP2 and CP11 had the lowest number of alleles. CP3 and CP6 the highest number of alleles. And then regarding the motif types, CP3, CP6, CP8, and CP10 exhibited compound motifs, with CP3 showing the highest. Conclusion: while rootstock vendors may sometimes offer sativa plants while ascertaining their hybrid cultivars, unfortunately, this study only identified one marker, CP4, that can be used to detect such cases of fraud in routine analysis on agarose gel. And so locus CP4 was the most significant in distinguishing cyanovent hybrid, showing bands exclusively in cyanovent samples, but absent in C. sativa. This confirms CP4's role in differentiating hybrids. However, a combination of CP4, CP2, CP6, CP9, and 10 primers without sequencing or capillary electrophoresis effectively distinguishes cyanovent from sativa due to their strong amplification and clustering specificity based in the phylogenetic analysis that I made. And then here you can see a publication of my paper down there with the citation and was published in Agronomy. Concluding remarks. Traditional plant species, I think, are important to be conserved because we need these plants. And particularly, I am coming from Africa, and I have never tasted or known what a chestnut looks like until I arrived here in Europe. And I think it's one of my best foods in Europe because I eat it all the time. I see that. Molecular identification methods are instrumental in facilitating the conservation efforts aimed at preserving the diversity array of these three species. Thank you very much, and that's the end of my presentation. And I would like to, if there are some questions that I need to address, or if there are some additions that some of the some of you can add to me in my career stage of research, I would be glad. Thank you.
Well, thank you very much, Dr. Tawfiq. Well, I would request that was a wonderful presentation from your side. I would request all the participants, if you have any queries or questions, you can ask now. All right. I think there is no questions from the participants. Thank you very much, Dr. Tawfiq, for your wonderful presentation. So let's move on to the next presenter, Dr. Falik Wahiba. Are you ready for the presentation? All right. So let me make you the presenter so that you can share your screen.
Okay.
All right.
So
So I did make
I
presenter. Please share your screen. Yeah.
okay. I start by present myself. I'm a
Yes.
research and educator in food science and biotechnology. I'm a degree in natural and life science, nutrition, agro-food, technology, and food science from University of Brother Mentori Constantine One. And my PhD in biotechnology from National High School of Biotechnology. I'm a Dr. Falek Wahiba. I have 40 years of teaching experience, specialty in food characterization, biochemistry, and analytical chemistry. Research include food biotechnology and wild plant ecology with notable publication on corn germ valorization and wild olive conservation. So, my affiliation in the laboratory of agro-food engineering, team of food process engineering, biodiversity and agro-environment in Ata Brother Mentori University Constantine One in Constantine, Algeria. So now
Dr.
now
Falik,
I
sorry
yeah
to interrupt you. I make you the presenter now. Could you please share us
yeah
your screen? Yeah, you can share
yeah
us the
I
screen
did
and you can.
I
Yes,
passed for my presentation.
yes,
So I try okay.
yes.
I you look my presentation.
Yes,
You look
yes,
just a
yes, sure.
minute. Okay. You look my presentation now. So I
Uh,
start. I have
yeah,
you look my presentation.
no, we didn't see that actually.
You not You not see my presentation?
No, no,
Now?
no.
No.
Just open the PPT and share. Yeah.
Active. Wait.
Yes.
Yes, I now it's okay. I think you
Yes.
look
Now
my
we.
presentation. Yeah.
Yes. We can see your
Okay.
presentation. Now we can start your presentation. Thank you.
Okay. I have
Yeah.
the honor to present to you our anti-flight assessment, assessment of the chemical antioxidant and antimicrobial properties of various solvent extract from wild olive leaves, Olea europea, Europea versilicis. In introduction, Algeria's flora includes a diverse range of medicinal plant species, many of which are utilized in traditional local medicine. Recent studies have highlighted the ecological significance of medicinal plants used in Algeria for both preventive and therapeutic purposes. Reasons for their use are low cost, availability, and average toxic effect compared to conventional therapy. While olive tree, Olea europea, Europea versicollis is a persistent aromatic shrub belonging to the Oleaceae family within distributed. Since ancient Greek time, leaf extracts from wild olive have been used primarily for their anti-inflammatory properties and treat various illnesses. Leaf extracts show strong anti-diabetic and cholesterol-lowering effects and may also possess antimicrobial properties. Their main objective are to determine the phenolic compounds and the antioxidant activity achieved by two methods of access ABTS and PPH. In addition, antimicrobial extract of different solvents from wild olive
Sorry to
leaves.
interrupt you, Dr. Faleh. Well, your screen is not moving. You're on the first, still on the first page.
Sorry,
Your screen is not moving. Are you still on the first page?
you look my presentation.
Only the first page. Are you on the first, still on the first page, or you moved it to the next page? Are you still at the first page? Dr. Faleh, am I audible? I'm unable to hear your voice. Could you please change the screen, the pages? Dr. Dr. Faleh, are you able to hear my voice? I
I'm
think
sorry.
that's all right.
I'm
That's okay.
sorry.
No worries. Yeah, we can hear your voice. Yeah. Yes.
Please, can you give me? I can.
Yes. Yeah. I'm giving you the
Okay.
access. Yes. I've already
Yeah,
done the make
thank you.
presenter.
Thank you.
Yes. Yes. You
Yeah.
can share the screen. Yes.
You look my presentation
Not now.
and now you look
Yeah,
it.
yeah. I'm just checking. No,
No.
no.
Okay. Okay. I try again.
Yes,
Problem of connection. I'm
yes.
sorry.
We understand. Maybe the problem with the
Yeah.
connection. Yeah, you can just try now.
Now you
Yes,
look my presentation.
yes. We look your presentation, but this is not the one.
You look it.
Yes, but this is not the presentation. This
You
is the
look
schedule.
it.
Yeah, yeah.
Wait. Sorry. Alors, and now
Yeah, I'm just checking. Open your PPT presentation, the PowerPoint.
yeah, I open it.
All right.
Now look my presentation.
No.
So I doing again.
Yeah, please
Now
do it again.
yeah, now it's okay.
No.
No,
No. Yes.
it's
Yes.
okay. Yeah.
Yes. So it's
In
okay. All right.
material method, we use GPS. We
Okay.
use GPS for collection of geocoordinates from sites in current connection. After harvest, samples were immediately transported to the laboratory, washed and dried. After that, the plant material was ground and stored until they were used. Maceration is the technique which consists of heating a concentrate of plant remain cold in organic solvent by extracting biosoluble quantities. Subsequently, the residue is concentrated by evaporated and then lyophilized until all traces of water are removed. The extraction of material using Soxley equipment using organic solvent, it significantly reduces extraction time by using two phases with final recovery of the solvent used. 20 of leaf powder was extracted with 5 solvents of different polarity and apolarity for 6 hours. You listen to me?
Yes. Yes, Dr.
Okay.
Falik. Wait, wait. Yes. Please,
Okay. I
please go on.
continue. Okay. I'm
Yeah.
sorry.
If
First
you're on the
graph
same
of
page,
leaf.
no worries. If the page changes,
Okay.
please try to change the page. All right. Okay.
Okay.
All right.
For
Please go ahead.
phytochemical screen of extract, it is a chemical technique that allows to determine qualitative to the different chemical groups contained in the extract by identification reaction of each chemical group. We tested the 4 solvent system 14 films liquid chromatographic illusion revealed by UV light. The total polyphenol were determined by spectrophotometry using the colorimetric method using 41 CO2 reagent and sodium carbonate by the following protocol. A calibration curve was performed in parallel under the same operation condition using gallic acid at different concentration. The concentration of the total polyphenol is calculated from regression equation of the calibration curve. The quantification of flavonoid was carried out by a method based on formation of very stable complex between aluminium chloride and oxygen atom present on carbon 4 and 5 of flavonoid.
Hello, Dr. Falik. Wahiba. Your voice is not audible. I think there's a problem with the internet. Dr. Falik. Yeah, yes, we can hear. Yeah, we can hear you.
Equation.
Please change the screen. I mean the pages. Are you
Yeah, I
still on the
changed.
first? Yeah.
You
Can
look
I,
it
yeah, we can see your
or
the page presentation of sample origin. Are you on the same page?
no, I'm in evaluation of antimicrobial activity. For
Then change it.
determination,
Ch-
yeah, I
All right.
change it.
But we can see only the first page, presentation of sample origin.
You look just the first page. For why? I
Change the
don't
slides.
understand.
If you're at
I
the
change it.
okay. All right. Maybe
I
there's a
don't know what
problem.
is the problem.
All right. All right. Then no worries. Maybe there's a problem with the connectivity. You can go on. Yeah.
Yeah, now it's okay. For the determination of antimicrobial activity in vitro, the disk division method, according to the protocol of traversy, the measurement of the diameter of the inhibition zone surrounding in the disk containing the samples to be tested was carried out in order to check the sensitivity of microorganism. Our study included 6 bacteria pathogenic to gram-positive and gram-negative. Staphylococcus aureus, Bacillus cereus, Pseudomonas aeruginosa, Escherichia coli, Enterobacter, Colicia, and Klebsiella pneumonia. In result and discussion, for the determine the figure present, the figure it's the same problem. I don't know for why. Repeated in the again. The figure present the extraction yield of extract of different solvent. The three extract methanolic, hydromethanolic, and ethanolic represent the best extraction yield between 60 and 65%. Subsequently, the average values noted for chloroformic extract, acetone and hydroacetone and ethyl acetone. For the result of phytochemical screening revealed a high concentration of secondary metabolites in the leaves of plant studies: flavonoid, quinones, glycosides, coumarins, terpenes, and saponins, and a total absence of alkaloid in all samples. Hydroacetonic and methanolic extracts showed better extraction with greater quantity of the different chemical families compared to the other solvents. The figure present the figure the figure presents the chromatography of extract for solvent solvent system pre and post UV visualization. We observe that it is migration and separation of green, blue, yellow, and brown spots with the second and the third system before the regulation and UV light for the hydromethanolic, hydroacetonic, and methanolic, ethanolic, hexanic, acetonic, deuterium ether, and ethyl acetate extract, and a complete absence of chloroform extract. The wild olives are very rich in phenolic compounds ranging from 174 to 10 equivalent acid gallic by gram of extract. The ethyl acetate, ethanolic, and methanolic extract of subtly following absolutely the hydroacetonic and hydromethanolic maceration. Subsequently, the hexane extract, deuterium ether, and acetone. For the concentration of flavonoid regulated the leaves of wild the leaves of wild are very rich in flavonoids from 2050 to 549 milligram of equivalent of quercetin by gram of extracts. Successively extracts ethyl acetate, ethanolic, and methanolic have the highest values, followed by the other extract, who have shown an apparent ability to extract flavonoids. The for the DPP, the antioxidant power proportional is proportional to the concentration and the percentage of inhibition increasing at increasing concentration of the extract, and the most potent antioxidant activity were identified with ethyl acetate and ethanol extract, which represent the highest percentage of anti-radical activity of ascorbic acid, followed by the methanolic and hydroacetonic extract. According to the data showing in figure 21, and by comparing the octane values with those of the standard, it is observed that the antioxidant activity of ABTS is proportional to the concentration. The percentage of inhibition increases with increasing concentration of extract. The diameter of inhibition obtained range from 0 to 32 mm on the strain, and the extract tested subsequently. The largest inhibition zones are observed with Staphylococcus aureus. However, the Klebsiella pneumonia is resistant to all extract at this concentration, making it the most resistant strain among those tested. In conclusion, and propose in conclusion and propose this this study demonstrates that using different solvents into extraction process significantly affects the total phenolic content, flavonoid level, antioxidant activity, and the concentration of secondary metabolites in wild oil leaf extract. Extract yield vary based on the method and the solvents used. The highest yields were obtained using methanolic and ethanolic between 60 and 65%. Moderate yields were observed with chloroform, acetone, hydroacetone, and ethyl acetate. The lowest yield come from hexane and deuterium ether. Biochemical analysis showed a notable presence of flavonoid, tannin, and sterol, along with formalin, polyphenols, glycosides, and saponin. Alkaloid, however, was not detected in any of the solvent extracts.
Dr. Falek Wahiba, I'm unable to hear your voice. Dr. Fale, are you able to hear my voice? We cannot see the screen. Please share us the screen. All right. Are you on the 36th page? We cannot hear your voice. Dr. Fale, maybe some technical problem over there. We'll wait for some time for Dr. Fale. All right, so maybe there's a problem with the internet connection. So let's move on to the next presenter. Dr. Vijayan Gurumurthy, are you available? Are you ready with your presentation?
Yes, sir.
All right. All right. So please introduce yourself and you can start off your presentation. Let me make you the presenter.
Uh,
All right. You can share your
can you
screen
see?
and you can, yes, introduce yourself.
Is it okay?
Yes, yes, we can see the screen. Yes.
Yeah. Good afternoon.
Good
Myself,
afternoon.
Dr. Vijayan Gurumurthy here. I am the presenter on the strategic environmental assessment process for the agricultural and horticultural process. And this particular presentation has been made based on my PhD research. Myself, Dr. Vijayan, who has published more than 400 research papers in leading international and international conference proceedings, national and international journals, and editor of more than 70 books, as well as abstract books, international levels, around 60 to 65. My H index is 60. So with this brief introduction, I would like to move my PowerPoint by uh, you know uh, starting with the strategic environmental assessment process for agriculture and horticultural projects. And I am thankful to CTP Forum for giving me an opportunity to present my paper. Am I audible? And am I visible?
Yes, yes, it's audible. You're audible, and but while speaking, it's coming as a resound. So
And
is it the speaker is
and
on? Okay, your voice is coming as a resound.
okay.
Yeah,
And what about my PowerPoint?
yep.
Is
We
it
can.
moving?
Yes, it's visible. We can see your presentation.
Moving.
Yes, please move on. Yeah, thank you very much.
So this process approach model is presented in this slide. What is process approach model for the sustainable agricultural and horticultural for effectiveness and efficiency? So you can just see that resources transformed into output products in efficiency as well as effective. Effective is quantitative in horticultural and agricultural projects, and efficiency means qualitative, the degree of excellence, the defects free from the agricultural and horticultural sustainability. The environmental impact assessment has been proposed. Environmental health impact assessment is a systematic identification and evaluation of the potential environmental health effects of the proposed projects, plans, programs, legislative actions with respect to the physical environment, chemical environment, biological environment, social economical environment of the total environment. So the organizations have to consider the sustainability as well as the environment in our organizations. But generally that is not taken place
Sorry
because
to interrupt you, Mr. Gurmati. We can see your only the computer screen, not the slides. So could you please share us your slides?
yeah, 1 minute, sir.
Sure,
I'll just arrange.
sure.
Or can you please take my PPT and roll? Can you take my PPT and roll?
Dr. Vijay,
Yes,
yes, you can share us your screen.
1 minute. I will explain.
Yeah, sure. It was everything fine, but the pages was like suddenly it goes off.
Is it my PowerPoint visible?
No, no.
Oh, is it my PowerPoint?
No.
No? Yes, yes. Now we can see your presentation. Yes. While
Okay,
giving the presentation, you're into the next page, then you can change the slides. Okay?
okay.
All right. Yeah. Please,
Environmental
please go
impact
on.
assessment process is proposed. But most of the agricultural and horticultural projects in Europe as well as other countries, this environmental impact assessment is not generally appreciated. Why? Because they are concerned about the economical as well as the social aspects, technological and economical aspects. This environmental impact assessment has been considered very important in my PowerPoint presentation. So the environmental impact assessment has air environment, surface water environment, soil and groundwater environment, noise environment, biological environment, cultural environment, and architectural, historical environment, archaeological environment, visual environment, and socio economical environment. Of the total 11 environments, and in these 11 environments, we may see the environmental impact assessment has to be conducted case by case, and the plus factor and minus factor are considered in project planning and decision making process, and public participation has to be also considered. For example, you take this particular grape garden. Grape garden was heavily affected due to the, this called the smelting plant dust fumes, and the gardens were affected in California, America. And what happened? This fumes have reduced around 80 to 90% of the global production and grape production, and the companies were closed. The grape production, wine production, manufacturing industries, they have closed the industries, and it was very important to control the air pollution. So the air pollution was controlled by the back filters, and these back filters finally has reduced the air pollution, and again the production of the grape in California gardens were again restored, and the wine production distillery industries have been started open. So this way, the production as well as quality has been implemented in project planning and decision making process by considering environmental health impact assessment. So environmental health impact assessment is systematically carried out to consider organizational planning and decision making process for the various physical environment, chem- chemical environment, biological environment, biochemical environment, and radioactive environment, as well as the man-made environment like cultural environment, as well as the social economical environment of the total environment. So we have two environment major. One is called the natural environment, biophysical. Another is not natural. It is a man-made environment or social economical environment. So this way, we will be able to conduct effectively and efficiently the environmental health impact assessment for the projects, especially the horticultural projects as well as agricultural projects, and the stress strain curve, and the environmental pollution prevention safe methods in agricultural, and root cause analysis, lack of organizational focus, mainly the lack of organizational focus concern about the three E's: engineering, economics, environment. Only two E's have been considered. Most of the projects in USA, UK, and other countries, European countries, why the productivity or the quality have been inferior, because they do consider in organizational planning the focus is mainly on economical as well as the social, engineering and economical. But the environmental impact assessment is proposed in this project. That is three E's: environmental health impact assessment, climate impact assessment, weather impact assessment. Alpha, that is alpha means zero point 0.5 degrees centigrade per watt per meter squared, zero point 0.7 degrees centigrade per watt per meter squared, climate sensitivity factor, and the climate net radiating force, that is zero point 0.7, 1.5 to 2.8 watt per meter squared per degree centigrade. So both are important in climate impact as well as weather. The change in weather conditions, that is the state of the variations in temperature, wind, rain, etc. So the business of responsibilities and sustainability is the CO2 emissions, greenhouse gas impact assessment, the environmental impact assessment, energy savings, the Streeter-Philip oxygen sag. So how the oxygen sag has been considerably affected on the eutrophication problems. Bloom's taxonomy, Plan Do Check Act for the agricultural and horticulturalist. So Plan Do Check Act. Plan Do as per the agricultural plans, horticultural plans as I have proposed. Do as per the plans. Check whether any deviations are there. Act based on the plans, and the action must be reviewed continuously. So it is a continuous improvement process. Circular economy, the life cycle assessment, and the life cycle assessment which will quantify as well as the qualify the environmental impact of the product and process from raw materials acquisition through end of life disposition. And waste treatment, the various waste treatment technologies like primary, secondary, tertiary treatments are proposed. The circular economy for agricultural and horticultural. For example, the waste treatment primary, secondary in India, but tertiary treatments are not available because that will have effects on the life cycle assessment, and especially the agricultural and horticultural projects. So the primary, secondary, and tertiary all should be completed in a input stage as well as the output stage, which is called process approach model. The previous slide I told about the process approach model. Life cycle assessment for the agricultural and horticultural process. 3R: recycling, reusing, and recovering stages. Solid and hazardous waste management. Solid waste management as well as the hazardous waste managements are proposed. And the rubberized cotton fabric rollers, which are used in cotton roller ginning industries in India. And crown corrosion in wastewater sewer pipelines. So most of the sewer pipelines have considerable impacts. That is called the crown corrosion. The corrosion take place on the surface of the sewer lines, and that will have effects like wastewater gas, like methane, hydrogen sulfide, ammonia, carbon dioxide. So which have considerable climate sensitivity factor of alpha nearly zero point 0.5 to zero point 0.7 degrees centigrade per watt per meter squared. So which have considerable impacts on the temperature, increase of the temperature and decrease of the dissolved oxygen in atmosphere. And that's why the sewers have to be properly designed and developed, right? And the noise pollution control. So noise pollution also is very important. Synergistic effects, and the toxic substance, how it will have the mortalitical effects on human health. and the human anthropological activity I is equal to, that is impacts, environmental impacts as well as the health impacts, population multiplied by affluence multiplied by technology multiplied by environment multiplied by sustainability. And the eco-friendly rubberized cotton fabric rollers is agricultural project which is proposed here. So the red color you see, this is the chrome composite leather clad rollers which are used in cotton ginning industries. As soon as I propose this project, and this require the eco-friendly models. Why means because the CCLC rollers have the adverse impacts on the environment because of chromium pollution, unsafe chromium pollution. The seed cotton, the double roller ginning machines, the lint cotton, white gold from roller ginning industries, the seeds obtained from the cotton ginning industries, the baling press. Now this is the laboratory model gins, and here the lint cotton is obtained from the seed cotton. See, this is a chrome composite leather, and it is due to the dust producing grinding and the chromium trivalent and hexavalent contaminates the cotton lint in the seed cotton industries in industries. This is a continuously dust producing the rollers. The rollers are being used in cotton ginning industries, right from 170 mm to 140 mm. 30 mm is the wearing rate for 3 months season. And chromium toxicity effects on human organs, dermal, another is oral, lung effects, and esophagus, as well as the absorption in the stomach, lung, and the dermal, oral. So this way, the chromium has considerable toxic effects on human being in our country, especially the occupational health effects, those who are working in the cotton ginning industries. The study of chromium-based disease as per epidemiological evidences, acute effects as well as the chronic effects. So ginning point is nearly 51 to 173 ppm as against the safe limit of zero point 0.1 ppm. And this is a single roller ginning machines, and this is a close-up of the double roller ginning machines, how it is ginning. And after 3 months, having about the 40 kilo of lint cotton, 45 kilo of lint per hour obtained from 130 kilogram of seed cotton put forth in the hopper. So the one third we obtained the lint cotton, and similarly the chromium pollution, it is continuously contaminating, dust producing grinding, and this have the contaminations of the lint cotton, just like water contamination, contamination. So this is called material contamination. So that is called a negative logarithm of lethal dose 50% test organisms in kg per kg in poison number, worn out and consumed washers discs after considerable period of usage for about 3 months. Dust producing, and dust have considerable impacts. So these are all agricultural, horticultural values have deteriorated because of the non-eco-friendly methods in technology wise are called greenhouse gas emission technologies, and they are not sustainable and environmental friendly, as we have seen the noted the environmental impacts is equal to PAT, population, affluence, technology, Technology has environment as well as the environment, social as well as the sustainability effects. For the sustainable development, the development that meets the needs of the present generations without compromising the ability and efficiency of the future generations to meet their own needs in horticultural as well as agricultural projects, plans, programs, legislative actions, and this worn out and air pollution. The people get affected in the vicinity, physiological as well as psychological effects. Synergistic poisoning, how this affected cross functional, the process approach and product approach. So product approach is only vertical. Two is engineering and economics, and three is means engineering, economics, and environment. Cross functional, it is from the left to right. So vertical means and horizontal. So occupational health worker, how they get affected. Chromium pollution analysis in cotton ginning industries. So the how lint cotton production that taken place in cotton ginning industries in India, and how the dust producing grinding rollers, inferior quality rollers, they are not superior quality rollers as designed and developed for the walrus in 1800s, and the USA, that is United States Department of Agriculture, has directly innovated the rubberized cotton fabric rollers, and the air pollution levels, SPM, RSPM levels are measured and monitored. GRED, gin roller experimental device with RCF as well as CCLC rollers. RCF means eco-friendly and CCLC means conventional method. The state-of-the-art methods, battery of plants, and new rollers drawings, how the innovations taken place. The roller ginning process is in process. The investigations are going on. The lint cotton, cotton technological parameters, fiber, yarn parameters, as well as dye catching properties. The economical status also concerns, though the new washer is costly, about 7 times more than the presently used CCLC rollers, but the RCF rollers is safe as far as the sustainability and environmental factors, which should be there in the sustainable agriculture and horticulture. So, as a conclusion, I would like to say that eco-friendly methods, greenhouse gas emission control technologies, and the old methods, conventional methods, which are all having effects on environment, need to be outdated for the sustainable development of agriculture as well as the horticultural projects. So the strategic environmental process that included the various organizational principles, because basically the organizations will be responsible for implementation of the various greenhouse gas uh, prevention technologies, and the new technologies for the new sustainable generations. And with this, I would like to elucidate. I have published 450 publications, citations 5000, H index 60. So the strategic environmental assessment process has been completed for cotton research. Reference is IEEE publications he cited, which has been published in 2018. And thank you very much for giving me an opportunity. C2 forum. Thank you, sir.
Well, thank you very much, Dr. Vijayan. That
Thank
was
you,
an
sir.
Yes, that was a wonderful presentation from your side. Well, I would like to ask the participants if you have any queries or questions, you can ask Dr. Vijayan now.
Thank you, Dr. David.
All right. I think there is no questions. Well, thank you very much once again, Dr. Vijayan,
Thank you.
and thank you. So let's move on to the next presenter. Dr. Falik Wahiba, are you ready with your presentation? Because you missed out with the presentation halfway. Dr. Falik Wahiba, are you able to hear my voice? Hello?
Hello, I listen you, but I have
Yes.
I want
Will you
to
be?
finish
Yeah,
presentation.
I can hear your voice. Will you be able to present now, or you need some time?
Uh, I just try. I try. You give me the
You try.
I can give. I cannot. You look my
Yes.
presentation for look my presentation, please.
Yeah, sure. Let me share the screen. I'll make you presenter. You can share us the screen.
Yeah,
Yes.
yeah, I
Yeah.
can now.
Yeah. You can
Yes.
just try now.
Uh,
If it is not possible, then no worries. Without slides, also you can give the
because I
presentation.
have this
Yes.
problem of connection.
Yes.
Now we will my presentation.
Yes. Yes. I did.
I
I did
continue
make
the
presenter.
completion.
Yeah.
Yeah,
Please continue. Yeah. Please continue.
yeah. Thank you.
Yes.
Using the
All right.
DPH method, the ethyl and ethanolic extract show the strongest antioxidant activity, while the methanolic and hydroacetonic extracts were with moderate effectiveness. In contrast, the acetonic, ethanic, ether, chloroformic, and hydromethanolic extract demonstrate the weakest activity. The methanolic, ethanolic, and aqueous extract of Olea orea versus obtained using three different methods. So the highest phenolic content and strongest on-activity in DPPH and IBTS acids. This extract may serve a natural antioxidant for reducing or prevention oxidative stress. All solvent extract share antibacterial activity against most bacteria with inhibition zone ranging from 0 to 32 millimeters depending on the strain and extract. Methanol, ethanolic, potassium, ethyl, and ethyl acetate extract were the most effective indicating that solvent significant effect on microbial potency. In prospective, the analysis of chemical compound of, obtained extract by high-performance liquid chromatography, assessment of antifungal effectiveness, and assessment of the anti-inflammatory effect of the perfect extract. And a comparative study of different wild olive extraction made on evaluating yield, antioxidant property, and essential oil composition. Thank you for your attention. Thank you for your attention.
Well, thank you very much. You're done with your presentation, Dr. Fale?
You listen me?
Yes. Are you done with your presentation?
I'm not understand you.
Yeah. Have you completed your presentation? Are you done with your presentation?
You want I repeat it?
No, I mean to say, ask you, are you have you completed your presentation or
Yes,
is still there?
yes,
Uh,
I think
all right,
I finished
because
the conclusion.
yeah, all right. Thank you very much. Because the screen was not moving, the slides was not moving. All right, no worries, Dr. Farik Waiba, though it was incomplete and you're completed now. You went away the middle of the session, and thank you very much for coming back and giving the presentation. Thank you very much. It was a wonderful presentation. I would request the participants, if you have any queries or questions, you can ask Dr. Falik. All right. I think there is no questions. Well, thank you very much, Dr. Falik Waiba. Let's move on to the next presenter, Dr. Fatma Hamada from Egypt. Are you ready, Dr.
Hello?
Fatma? Yes.
Yeah,
All
yeah.
right. So I'll make you the presenter, wherein you can share the screen. Please introduce yourself, and you can start your presentation.
Yeah, hello. It's a pleasure to be among you today. My name is Fatma Hamada. I am a lecturer in Aswan University, Egypt. Okay. My study about ethnobotany. We will take presentation.
Yes.
Yeah, I'm
Dr.
trying.
Fatma, yes, I've given the access. I already marked the make presenter. You can share the screen now.
Yeah, I'm trying to upload it.
Yes.
Sorry, it's first time here to with this program. With Zoom, it's easier, but where I shall open. I think the file. Okay. Yes. Yes, let's begin. Okay. Okay. It is concerned with taxonomic diversity of medicinal herbal plants used in northern
All right.
community and Saudi Arabia. Okay. Uh, as we
All
see
right.
about study
Dr.
area.
Fatma, yes, we can. Yes, we
Yes.
can see the presentation slides. Please go on. Yeah, you can start your presentation.
Yeah. Okay. About the study area, it is located within Ha'il province. Okay, or department, and it is in the northern part of Saudi Arabia, central north. Okay, here it is surrounded by many places like Al Jof, okay, Tabuk, Al Medina Munawara, and Al Qasim.
Dr. Fatma, are you there? Well, maybe there's a problem with the internet connection. Let us wait for some time. Dr. Fatma, are you there? Maybe there's some technical problem. We'll wait for some time. Yep. Yes, maybe there is a problem with the internet connection with Dr. Fatma. So let's move on to the next presenter. Dr. Gulmeriza Hidari, are you available?
Yes, I am ready.
Are you ready
Hello,
with the
dear
presentation?
Doctor
All right, all right. So
David.
let me make it. Yes, let me make you the presenter, and you can share the screen, and you can start
Thanks,
your presentation with the introduction. All right.
thanks.
Thank you very much.
Bye.
All right, please start now.
Dear Dr. David, do you have my presentation?
Yes, yes, we are able to see your presentation. Yes,
Thanks.
you can start off directly. Yes.
Thanks. Hello, everyone. I am Ghulam Reza Hidari, faculty member, associate professor at University of Kurdistan in Iran. I work in the field of crop ecology. I would like to express my sincere gratitude to the organizers of the conference for giving me this opportunity. The title of my presentation is Enhancing Quinoa Yield and Quality Through Different Levels of Irrigation and Vermicompost Application. Quinoa, Chenopodium quinoa, is a highly regarded cereal from the Amarantaceae family, recognized for its nutritional value and versatility. It is gluten-free, rich in essential amino acids, antioxidants, and offers a protein content of 14 to 20%, making it a beneficial food source for vegetarians. Efficient agricultural practices like deficit irrigation and high-efficiency systems such as drip irrigation are crucial for optimizing water use and enhancing crop yield. Khaled et al. And Fatemi-Kian et al. In two separate experiments showed that full irrigation produced the highest seed and biological yields of quinoa. Vermicompost, a nutrient-rich organic fertilizer produced through the decomposition of organic waste by earthworm and microorganisms, provide essential nutrients for plant growth. Vermicompost application has shown positive effects on quinoa yield and quality under various conditions. It can enhance biomass production and growth status of quinoa under drought stress. In drip tape irrigation systems, vermicompost application of 15 tons per hectare resulted in the highest quinoa seed yield and improved soil properties. Objectives of our study. The primary objective of this study was to evaluate the effects of different irrigation levels and vermicompost application on grain and biological yield of quinoa to promote sustainable agricultural practices. The research aimed to determine the optimal combination of irrigation and vermicompost that enhances quinoa and nutrient concentrations. Now, I want to talk about materials and method were used in this study. Site description. The study was conducted in the agricultural research field at Tehkolan Plain in Kurdistan province, Iran, during 2021 and 2022. The region experiences an average annual rainfall of 350 millimeters, with maximum and minimum temperatures of 23 centigrade degree and 60.6 centigrade degree, respectively. The soil analysis of the experimental farm indicates a clay loam texture, a field capacity of 35%, and electrical conductivity of 0.49 decimens per meter, a pH of 7.62, and organic carbon of 0.76%. Experimental design. A split plot experiment based on a randomized complete block design was employed, with 3 replication over 2 years. The main factor included 3 irrigation levels at 50, 75, and 100% of the quinoa crop's water requirement. The sub-factor involved 4 levels of vermicompost fertilizer at 0, 5, 10, and 50 tons per hectare. The quinoa variety Titica was selected for its adaptability to mountainous regions. The experimental plots measured 4 meters in length and 3 meters in width. Irrigation system characteristics. A drip tape irrigation system was utilized. Maintaining a 7-day irrigation interval, soil moisture was monitored using a volumetric meter and soil moisture sensors. The depth of irrigation was calculated using the soil moisture balance method. The traits were measured in this study were grain and biological yield, seed nitrogen content, seed phosphorus content, seed calcium and potassium contents, and seed oil percentage. Now, I would like to talk about result and discussion. The grain and biological yields of quinoa was significantly influenced by both irrigation and vermicompost levels, although their interaction was not statistically significant. As you see, the table 1 shows mean comparison of irrigation and vermicompost effects on the grain yield and biological yield of quinoa in 2 years of experiment. About grain yield, we can say the highest grain yield of 1,987.70 kilogram per hectare was achieved with 100% of water requirement irrigation, while the maximum yield of 1,784.01 kilogram per hectare was recorded with 15 ton per hectare of vermicompost application. Vermicompost utilization significantly enhanced grain yield across all irrigation levels. About biological yield, as you see, the highest biological yield belongs to the full irrigation treatment. Furthermore, application increased biological yield compared to non-application, with no significant differences observed among vermicompost levels. Numerous studies indicate that water deficiency during the growth and seed filling stages negatively impacts biological and grain yield by hindering photosynthesis. Water stress, especially during seed filling, accelerates leaf aging, shortens seed filling, decreases average seed weight, and ultimately reduces yield. In contrast, stress-free conditions with sufficient water promote higher biological and grain yields by enhancing the plant source and sink strength, leading to increased seed yield. Augmenting biological and grain yield through increased vermicompost application can be achieved by promoting plant vegetative growth, enhancing reserve material production, improving floor fertility, and increasing thousand seed weight, ultimately leading to increased seed yield. The concentrations of phosphorus, potassium, and calcium in quinoa seeds were significantly influenced by both irrigation and vermicompost levels, although their interaction was not statistically significant. As you see, Table 2 shows means comparison of irrigation and vermicompost effects on phosphorus, potassium, and calcium concentrations of quinoa seed in 2 years of experiment. Higher concentrations of phosphorus and potassium were found at full or 100% irrigation of quinoa's requirement, irrigated compared to 50% and 75% irrigation levels. However, the highest calcium content of the seeds belong to the full irrigation treatment. Additionally, the application of vermicompost enhanced the nutrient concentrations in the seeds. The highest phosphorus concentrations, 0.36 milligram per gram, was observed at 15 tons per hectare of vermicompost. The application of vermicompost also significantly increased potassium levels compared to the control. Furthermore, calcium concentrations in seeds increased with higher vermicompost application. The concentration of nutrients in seeds increases with higher irrigation levels due to improved soil moisture, nutrient availability, and enhanced nutrient absorption by plants. Continuous soil moisture availability under drip irrigation helps dissolve nutrients near their roots, facilitating better absorption at higher irrigation levels. Studies have shown reduced phosphorus levels in quinoa seeds under stress compared to control treatments. It seems that vermicompost enhances plant access to essential nutrients, particularly phosphorus, by stimulating soil microorganisms and improving nutrient absorption. Its compounds acidify the rhizosphere, which helps release insoluble phosphorus as hydrogen ions are replaced by calcium ions. Since phosphorus is immobile in soil, plant roots can absorb it when they come into contact with organic or inorganic sources that contain in its available forms. The next slide shows the mean comparison of effect of different year and different irrigation levels and vermicompost fertilizer. Analysis of variance showed that nitrogen concentration in quinoa seed was significantly influenced by the year irrigation levels and vermicompost application, including their triple interaction. As you see, seed nitrogen levels increased in the second year, influenced by varying irrigation and vermicompost treatments. Analyses revealed that higher vermicompost levels corresponded with increased nitrogen percentages across all irrigation treatments. Furthermore, nitrogen concentration was greater in low irrigation conditions compared to full irrigation. The increase in seed nitrogen concentration with decreased soil moisture is due to the concentration effect, where plant growth slows more than nitrogen absorption, leading to higher nitrogen levels in seeds. Conversely, excess water can cause nitrogen loss through leaching, reducing seed nitrogen concentration. Additionally, organic fertilizers like vermicompost enhance nutrient supply, improve soil conditions, and promote root and aerial organ development, further facilitating nitrogen absorption. The study revealed that a significant interaction of irrigation and vermicompost on soil on seed oil content. As you see in table 3, mean comparison of effect of different levels of irrigation and vermicompost on quinoa and seed oil content. Seed oil content varied from 2.31% under 50 irrigation without vermicompost to 4.2% with 75% irrigation and 15 tons per hectare of vermicompost. The results showed a notable increase in oil percentage with vermicompost across all irrigation levels, while irrigation alone didn't enhance oil percentage in treatments without vermicompost. The oil percentage in seeds, a quantitative trait influenced by multiple genes, is negatively affected by drought stress, which damages these genes and reduces oil content. Our study supports earlier findings that water stress diminishes oil percentage by impairing the seed's ability to accumulate oil due to hindered absorption of necessary assimilates. Adequate water availability enhances seed weight and oil storage, while drought leads to the shorter seed filling period and reduced seed weight, further lowering oil content. The use of vermicompost fertilizer can improve root growth, enhance water and nutrient uptake, and balance soil acidity, ultimately increasing seed oil percentage. Conclusion. The study finds that vermicompost application under varying irrigation levels significantly boosts quinoa yield and quality, particularly in low irrigation conditions. It effectively mitigates drought stress impacts, which tend to lower yield. Vermicompost enhances water and nutrient absorption, improves soil conditions, and supports quinoa growth while optimizing water use. Higher irrigation and vermicompost levels lead to increased essential nutrients such as nitrogen, phosphorus, and potassium. The nutrient-rich vermicompost improves plant nutrition and soil properties, enhancing seed nutrient concentrations. The study advocates for using organic fertilizer like vermicompost to improve quinoa seed quality and soil health, especially in Iranian soils lacking organic matter. Thank you for your attention.
Well, thank you very much, Dr. Guram Reza Hidari. That was a wonderful presentation from your side. And I would request all the participants, if you have any queries or questions, you can ask now. Well, I think there's no questions. Well, thank you very much, Guram Reza Hidari. Thank you very
Dear
much.
Dr. David,
Thank you very much. Let's
dear,
move on to the next
dear,
presenter.
dear,
Um,
Dr. dear Dr.
sir,
David, I
yes.
appreciate the opportunity to share my research with you all, and I welcome and I thank you for anything.
All right, thank you very much. Thank you once again.
Thank
So let's move on to the next presenter, Mr. Carlos Alberto from Chile. So are you ready with your presentation, Mr. Carlos?
You?
Yes, Mr. Carlos.
Yes,
Are you
yes.
all right? All right.
Can you hear me? Excuse me.
Let me make the presenter so that you can share your screen. Yes.
Yes.
All right.
Ah, Dios mío. For sharing the. Ahí está.
Yes, we can see the screen. Now share us your PowerPoint presentation.
Yes, yes.
Yes.
Can you see the presentation?
Yes, yes, Mr. Carlos, we can see your presentation. Now, please introduce yourself and you can start off your presentation. All
Uh,
the best.
how can I?
Yes.
How can I? The presentation is too small. How can I? hmm, how can I expand the presentation?
Well, at the bottom of the page, you can see the red button.
Yes, yes.
Click
The
on
red
that.
button.
Yeah, just
See,
click on that. Yeah, click on that.
see.
I'm sorry, on the top of the page,
Yes,
the red button. Just
yes.
click that. Just click that.
Can you see the presentation?
Yes, yes, we can see your presentation. You can go ahead. No, no worries. You can go ahead. Yeah.
But it's too small for me.
Yes. So can I help?
Yes, please, please.
Okay. At the bottom, just at the bottom of the presentation, you
Yes.
can, yeah, you
Where is
can
the
see
bottom?
the bottom at the right hand corner. In the right hand corner, Y- can you kindly take your cursor to the right hand
On
corner?
the right,
Yes.
yes.
Yes. There is like a human being I can see in that part of. No, go down. Just no.
Plegar.
Yes. Yes. On that tab where you have
Yes.
the no. Come down with your cursor. No. The right hand corner. Yes. No.
Can
Bring
you
the
s-
cursor. Yes. Where you have a structure that is slideshow. If you put on the
can you
where
see
you
the
can,
presentation?
yes,
Excuse me. Can you see the present? I can see the presentation
where
now.
you can zoom. You can at the right hand corner where you can zoom. You can increase the size of the slide. Yes, you can see that there is one icon. Yes, up. Just go up a little bit. No, no, come down where you were.
Yes.
Yes, on that arrow with the minus and plus, you can see that next to the minus and plus, you can see slideshow. There is an icon just by the minus sign. You can see the minus sign.
On the right side.
Where the cursor is, there is a minus sign and a plus sign. Close to the minus sign, there is an icon. No, no. Go back to the left. Yeah, that place.
Yes.
Yes, you can see the
There.
minus sign. Yes, can you see the minus? Like the zoom where you can minus and
No,
subtract.
I can see the zoom. Excuse me.
Yes, subtraction. Yes, that one. There is
Here. Do you see
yes,
that?
yes. There is a plus sign. Plus.
Plus
Yes, just below your cursor, there is a plus sign,
here.
and where your cursor is currently at, there is a plus sign just below your cursor, and there is a minus or a negative sign below the cursor. No, come down. No. Yes, where your cursor is currently at, you can see in. Is it possible if you, yes, bring it down? Y- Yes. No, no, no. You don't. You're going up just at the right hand corner. Down. Yes.
Down. Yes.
Yes. Can you see the percentage?
I see that. Yes.
Yes,
Yes.
yes, we can see your
Yes.
presentation. Yes.
There's an icon just close to the minus. Yes, that icon. Yes. Click on that. Yes.
Do you see
All
the
right.
presentation?
Yes,
Yeah.
yes, the full presentation. Please go on. Yep.
Excuse me. Thank you very much. This study is titled comparative study on antioxidant activity and phenolic content in roots, leaf, and fruits of Cauteria mucronata. This research study was performed in Los Angeles City, in Chile, in the University of Concepción, in Spanish, Universidad de Concepción. The work was performed by the authors, by the speaker, Christopher Martinez and Eduardo Navarrete. Polyphenols, which are found in plants, are natural antioxidants that function as reductants, hydrogen donors, and singlet oxygen quenchers. In addition, these compounds have antimicrobial activity. Other activities of this kind of secondary metabolites are uh, anti anti-cancer activity, anti-obesity activity, inflammatory activity, and regulation of blood glucose and lipid in blood. Polyphenols reduce the risk of the development and progression of many human chronic diseases. And they have in common a polyhydroxyl structure, and this kind of structure is the basis of the biological activities of the polyphenols. The presence of phenols in plants is justified because abiotic stress, such as drought, leads to higher levels of polyphenols in the stressed plant. Other 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 abiotic stresses are include water flooding.
Well, sorry to interrupt you, Dr. Carlos.
Yes,
The slides is not moving. Are you still at the introduction page?
I don't understand you. Excuse me.
All right. Are you still at the introduction page? Because the slides are not moving.
Do you hear me?
Yeah, I can hear you.
Do you see the presentation?
Yeah, we can see your presentation. My question is, are you still at the introduction page?
Yes.
All
No.
right.
Yes. Yes. I am. Excuse me. This is the introduction.
All right. All right. Please go on. Yeah. Thank you very
This
much.
by way of introduction. By way of introduction, polyphenols are found in plants, and this the presence of polyphenols is in the presence of abiotic when the plant lives in the presence of abiotic stress like water flooding, low and high temperature, acidity and alkalinity of soils, and um, and radiation of the sun, high radiation, and the plant Gauteria mucronata was collected in the region Araucanía in Chile, South America, in Chile, in the Araucanía region, and in the Melipeuco commune, where around the Jaima volcano. Melipeuco is a village in the Araucanía region. The Jaima volcano, the plant was collected around the Jaima volcano, where there is a volcanic soil, and this soil has high water drainage and acidic soils, as an acidic soil. And this plant is able to live under this condition, under this abiotic stress. Gaultheria mucronata is a shrub native to Chile. It's a vascular plant, and its common name is chaura. Its geographical distribution is from Maule region to the Magallanes region in Chile. The length of the plant is 20 to 80 centimeters up to 2.5 meters. The length and width of the leaves is between 1 until 2 centimeters, and the width is 0.4 to 0.6 centimeters. The fruit of the diameter of the fruit is 6 to 8 millimeters. In this study, we we analyzed it separately, the fruits, the leaves, and the roots of this plant. As the the materials and methods were the following. As extraction procedure, with an extraction procedure, we obtain three kinds of extract: the methanolic extract of fruits, leaves, and roots in a socklet apparatus, and the extraction solvent was methanol. After the extraction, we proceed to evaporate the solvent in a rotatory evaporator, and after that we keep the extract in an oven until dryness. The the anti the the antioxidant activity, the assays for determining the antioxidant activity was performed with a mixture composed of 2 milliliters of radical and 165 microliters of the methanolic extract. The homogenized mixture was read in a spectrophotometer at the wavelengths that correspond to the wavelengths of the DPPH method and ABTS method. And the wavelengths were 517 nanometers and 734 nanometers. Before the measure, there was a 1 minute of reaction between the methanolic extract and the radical ABTS or DPPH. With the values of IC50 obtained from the graphs of concentration of methanolic extract versus percent inhibition of absorbance, a kinetic assay was developed. In other words, the effect of the antioxidant activity over the time. In this experiment, a mixture composed of 2 milliliters of radical and 175 microliters of the methanolic extract were homogenized, and this homogenized mixture was measured in a spectrophotometer every 5 minutes for 150 minutes at the wavelength that corresponds to the DPPH method and ABTS method. The total polyphenol content was determined using the following Sivocaltone methods with gallic acid as standard as a standard phenol, and the total flavonoid content was also determined by using catechin as a standard secondary metabolites or polyphenol at the wavelengths that correspond to these methods in a spectrophotometer. The concentration range for the experiments were different. We can for the roots, the concentration were not so high as in the leaves and as in the fruits. The level was a minor level of concentrations in milligrams per milliliters. The results were the following. The IC50 values or results were obtained from the graphs of percent inhibition of absorbance versus concentration of the different extract. All the experiments were performed in triplicates, and for calculating the IC50 values. As we can see in the graphics, when the concentration of the extract increases, there is also an increase in the percent inhibition of absorbance. We made experiments separately for methanolic extract of fruits, methanolic extract of leaf, and methanolic extract of roots. For the roots, the results show the results showed a minor level of a lesser level for the value of IC50, and we can interpret that as we can that means that the roots has a more antioxidant activity than fruits and leaves. We can see we can see these results in this graph where no significant differences. There were no significant differences between leaves and roots, although in the graph we can see a difference. The the antioxidant activity was also calculated as gallic acid equivalents expressed in milligrams of gallic acid per 1 gram of methanolic extract by the DBPH method. The roots show the results for the roots showed a higher content of gallic acid. As we can these results, we can see these results in this graph, where the fruits showed a minor level. The antioxidant effect over the time, over 150 minutes, was during the time over the time was similar for fruits and leaf for leaves and roots, and with the fruits take a longer time for reaching the higher level of antioxidant activity after 60 minutes. Oh, excuse me, I can't continue.
I think, hello.
See?
I think
Yes,
I think you need to use the arrow keys on your keyboard rather
I
than
can't
the
continue,
mouse.
excuse me.
You have to use the arrow keys on the keyboard, left and right, other than using the mouse. No, you should not use the mouse. I think you should use the keyboard.
I can't continue, excuse me.
You can try to exit with the escape key, and then you come back with the same procedure that we followed. I think I cannot hear him.
Doctor, you are not audible to us. Can you hear me? Yeah, you are not audible to us. You are not audible. Yeah, please check your mic, please. We are unable to hear you.
Do you hear me? The antioxidant effect of the metabolic extract over 550 minutes using the IC50 concentration showed a similar effect for all the extract. After and the higher level was reached after 30 minutes, the higher level of antioxidant activity. The total polyphenol content expressed as gallic acid per 1 gram of methanolic extract was higher in the root extract, as we can see in this graph. The total flavonoid content was also higher in the extract of the roots, and a minor level was observed in the extract of the fruits. Again, the roots show the higher content of polyphenols. as in the above mentioned experiments. As conclusion, the obtained results revealed that the tested species exhibited radical scavenging, and furthermore, the antioxidant activity of methanolic extract from roots of Gauterium grunata were found to be the most potent. These results could be considered as a first step for the research of biological effect of Gauterium grunata related to phenolic compounds. And as a first place, we can consider the roots, because the roots has demonstrate a higher content of phenols and a higher antioxidant activity. As acknowledgement, thanks to project of the Universidad de Concepción, and thanks to Mrs. Claudia Flores for her technical support. Thank you very much for your attention. Do you hear me? Excuse me, do you hear me?
Yes, I can hear you.
Yeah, Dr. Carlos Alberto, you're not audible to us. Doctor, have you done with the presentation?
Yes, I have finished the presentation. Do you hear me?
Yes, now I can. Yeah, please, Doctor. Yeah, thank you, Doctor. Thank you for the presentation. Yeah, any
Thank you very
questions
much.
to Dr. Carlos Alberto? Yeah, I think there is no questions. Then we'll move on with the next. Yeah, Dr. Sana, do you have a question? Dr. Falek Wahiba.
Do you have a question?
Yeah, Dr. Falek Wahiba, you are not audible to us. Do you have a question?
Excuse me, I'm waiting for the question.
No, Doctor. I think, yeah, there is no question. Yeah,
Thank you very much for your attention and for your help.
Hello, I have a question.
yeah, there
Yes,
is a question, Doctor. Yeah, from Taufik Saleh Yusuf.
Yes, I would like to know if the phenolic compounds that you selected, why did you select uh, Um, Sorry, just missed my question. Uh, what phenolic compounds were you looking at?
I'm looking for phenol compounds, for polyphenols, polyphenols compounds, because
Yes, polyphen.
compounds has various different biology by different activities and these activities are can help the human health, and they can and the extracts could be used as antifungal in infections that present in plants as a biopesticide.
Okay, all right. Thank you.
Flavonoids and phenols are important. They can use as biopesticides, and that was the reason.
Yes, so I wanted to identify the phenolic compounds that you observed during this study.
Excuse me, this is a first step of this plan of the study of this plant, because these plants, the studies are not are there are no studies of these plants. The studies are very
Oh,
few, are
oh,
small
okay,
studies. Hmm? And these
okay,
plants has a potential in the roots. Hmm? In the
all right,
roots.
okay. Thank you very much.
Thank you for your question.
All right. Thank you very much for your wonderful presentation. And let's move on to the next presenter, Dr. Sana Khalid. Are you ready with your presentation? Dr. Sana, are you able to hear my voice?
Yes, I could hear your
All right.
voice.
All right. So I'll make the
Yeah.
presenter now. You can share the screen and you can introduce yourself and start your presentation. All the best. Dr. Sana, please share us your screen. Dr. Sana, are you able to hear my voice? Dr. Sana Khalid, are you able to hear my voice? I think there is a technical problem. Dr. Sana Khalid, we'll wait for some time. You should join us. Please be patient. Dr. Sana, are you able to hear my voice? Hello? Dr. Sana, could you please share us your screen? I already make you the presenter. Your voice is not audible. Dr. Sana Khalid, are you able to hear my voice? If yes, please respond. I think there's some technical problem with Dr. Sana. All right, we'll take a small break and we'll come back after 20 minutes from now. And the next presenter will be Dr. Sana. Till that time, we can take rest. Thank you very much.
अगर थोड़ा लेट हो जाए तो बैठी नहीं यहाँ पे करके ही जाऊंगी। अगर थोड़ा लेट हो गई कि कनेक्शन आगे पीछे नीचे बंद है। चल मैं भी कुछ अल्लाह हाफिज़।
Hi everyone, Dr. Sana Khalid. Are you able to hear my voice? Dr. Sana, are you able to hear my voice?
Yes, I am
Just
watching and I
all right.
am waiting for a long, long time.
Yeah, it was a short break actually. I'll make you the presenter. You can share us the screen and introduce yourself and start your presentation. All right. Yeah, already I make the presenter. You can share us the screen now.
Okay.
I'm unable to hear your voice. Share us the screen, Dr. Sana. Dr. Sana, are you able to share the screen? I already make you the presenter. Just open your PPT and share us. Your voice is not audible, Dr. Sana. Due to some technical problem, Dr. Sana is logged off. Let's wait for some time. Mr. Rakibul Hassan, are you available? Hello, Dr. Sana. Are you able to hear my voice, Dr. Sana? Dr. Sana, we cannot see your slides.
Yes.
Yes, I've already make you the presenter. You can share the screen now so that
Okay.
we can see the presentation. Yes.
Can anyone see my slides or not?
Yeah, you can share us the screen so that you can see the slides and you can give the presentation. Even we can see this slides. So I've already make you the presenter. Just
Okay.
share your screen.
Should I start?
Just open the PowerPoint presentation. Dr. San, I'm sorry, we cannot hear your voice.
Their names and affiliations. What are the
Yes, we
research
can.
highlights? First
All
of
right.
all, the isolation and molecular characterization of the chickpea chlorotic dwarf virus from the symptomatic leaves of Lupus cylindrica. Then, in silico analysis of the chickpea chlorotic dwarf virus was performed, including the phylogenetic diversity and mutation analysis. And the third one is comparative metabolic profiling of the infected and healthy L. cylindrica plants was performed through the GCMS, that is the gas chromatography mass spectrometry, Fourier transform infrared spectroscopy, FTIR, and X-ray diffraction, that is XRD. Next is what is the introduction of this research. Lufa cylindrica, commonly referred to as the sponge gourd, and it is dichotomous plant vine species belonging to the Cucurbitaceae family that is originating in tropical Asia. The cultivation of the sponge gourd is subject to various biotic and abiotic factors, with virus-plant interactions playing a crucial role in influencing both positive and negative effects on plant metabolism and altering their metabolites. Among the biotic stresses, the virus that belong to the Gemini viridae family, the Gemini viruses that are exclusively transmitted by the whiteflies. However, some of their genera, they are transmitted by the leafhoppers, aphids, and tree hoppers. The evident symptoms of the Gemini virus infection include the leaf curling and crumpling, resulting in compromised plant growth characterized by the fewer plants and smaller leaves. A notable member of the Gemini viridae family is the chickpea chlorotic virus that belong to the genus Mastri virus that are transmitted by the leafhopper. The virus transmitted by the leafhoppers consists of the four open reading frames and a circular single-stranded DNA genome. In this study, the leaves of the L. cylindrica are showing symptoms of the chickpea chlorotic dwarf virus infection, and healthy one collected characterized on the molecular and basis and comparative metabolic profiling was done. Next is, what is the research problem? Lufa cylindrica is an economically important crop in Pakistan. It has been infected by the Gemini viruses and poses a serious threat to its productivity and significantly reduces its yield. While considerable efforts have been made to manage the disease, the impact of chickpea chlorotic virus on the plant's metabolites remains largely unexplored. Next is, what is research question? Does chickpea chlorotic virus lead to changes in the metabolic profiling of the Leaf cylindrica? Then another one is, what is the research hypothesis? Yes, chromatography, GCMS, FTIR, and XRD were used to analyze the metabolic profiles of the diseased plants and compare them with those of the healthy plants. So these are the objectives of the study. First is to characterize the chickpea chlorotic virus on molecular basis isolated from the Leaf cylindrica, and then compare and analyze their metabolic profile of healthy and infected plant samples. Then comes to methodology. First of all, the sample were collected, leaf samples were collected on the basis of their morphological features such as the leaf curling, crumpling, chlorosis, and they are packed in two different packets. One is healthy one and another one is infected one. Then they bring to the lab for the molecular characterization. DNA extraction was done by using the CTAB method, and PCR amplification of the chickpea chlorotic virus was done by using the universal primers, clone them, sequence, and blast NCBI for the chickpea chlorotic virus confirmation. And some of the leaf samples, they were washed and dried, and they were ground into the fine powder to perform the metabolic profiling. In between the two wet lab, there is a dry lab experiment, that is in silico prediction of the diversity mutation analysis of the chickpea chlorotic virus. First of all, the phylogenetic analysis was done by using the NCBI blast, muscle in mega 11 and maximum likelihood method, and then recombination was detected by using the RDP4 program into the aligned sequence of the mega 11. Then at the end, metabolic profiling of the virus infected and healthy leaves was done by using the GCMS, FTIR, and XRD techniques. Then what were the result? First of all, these are the sample. These are the pictures of the uh, virus infected leaves. These are the virus infected plant, and this is the virus infected leaf, and this is the healthy one. We checked as a control, and these are the experimental. Then this is the complete illustration of the mastery virus, that is the chickpea chlorotic virus, that is approximately 2600 pairs. These are the two conserved regions, and it has four open reading frames. The movement protein, code protein, rep A and rep both are responsible for the replication. The molecular characterization of the infected sample was done by using the DNA extraction, then using the PCR amplification, and we get these result. This is the 1 kb ladder, and this shows the 3 kb, and our virus is approximately 2700 base pairs. So lane 1 is the control plant, lane 2 and 3, they are the experimental or the disease infected plants, lane 4 is a negative control, and 5th is the positive control. Next is, this is the table of the virus, of the whole different host of the viruses. The chickpea chlorotic dwarf virus was isolated not from the chickpea, but it has been isolated from a range of other hosts, and that belong to the different families as well. Host range is broader than expected. Then in silico analysis was done. This is the phase 2 of the research. First of all, the phylogenetic analysis was done. 49 chickpea chlorotic virus genome was retrieved from the GenBank database, isolated from the varied variety of host plants at different years and different geographic location worldwide. These 49 viral genomes of chickpea chlorotic virus were assembled into a single dataset, that is the FASTA format. Then phylogenetic tree was constructed by using the nucleotide sequence of the chickpea chlorotic virus, and they were make clusters with the other virus isolated from this research. The sequences were aligned by using the Mercer Mega 11 software, and the phylogenetic tree was created by employing the maximum likelihood method by using the 1000 bootstrap value. This is the phylogenetic tree. This one is our virus that was isolated from the Lufa cylindrica, and it it resembles most with the cucumber virus that was isolated from Pakistan in 2016, and they belong to the same cluster, that is the Indian virus. Sorry, that is also the Pakistan virus that isolated from the Goossypium hirsutum, the cotton plant. Phylogeny shows that it belongs to the Pakistani strains, and it has expanding its host range from Gossypium hirsutum to from chickpea to Lufa cylindrica. The next is recombination analysis using the RDP. The RDP program was utilized to distinguish possible parents and the region of the recombination. So the above virus, it exhibit um, the recombination between the JF831147 strains, that is the chickpea chlorotic virus isolated in India in 2012 as the major parent, and MF1781119, that is the chickpea chlorotic virus strain, Pakistani strain, isolated in 2019 as the minor parent. So this is the result. Approximately 20 events have been detected by using the RDP program. These are the events, and this is our isolated virus, and its possible major and minor parents are depicted here. So this is the brief summary of the RDP program, which shows the alignment and the recombination sequence where the recombination was present, and major and minor parents. These are the programs. Approximate more than 3 plus constitute a positive program. And the next phase is the phase number 3 is the metabolic profiling of the chickpea chlorotic virus, and they are compared with the healthy one. This is the qualitative analysis of the compounds isolated from the healthy and infected L. cylindrica. 12 compounds were identified from the healthy plants, and 9 were identified from the infected plants by using the GCMS analysis. And they are quite different from each other. Then FTR analysis was done of the healthy ones, and they characterize the healthy plants, which evaluate different functional groups on different peaks. Healthy plant include alkenes, nitro compounds, alkane, and alcohol. Next is, this is the absorption group and class compounds which were isolated from the FTIR from the healthy ones. Then FTIR was also done from the leaves infected with the chickpea chlorotic virus, and these compounds, their appearance, group, and class were isolated from the FTIR analysis. Number 13, XRD, X-ray diffraction pattern of the healthy leaves of L. cylindrica, which indicate crystalline structures, well-defined peaks at specific angles, indicate an organized crystalline structure. And this is the result of the infected leaves of L. cylindrica. At last, at the end of the research, what is the conclusion of this all the research? The study utilized the CTAB method for the extraction of DNA from the healthy and infected plants and PCR amplification of the DNA region confirming the presence of chickpea chlorotic virus. After that, phylogenetic analysis demonstrated that chickpea chlorotic virus isolated from the L. cylindrica exhibited close genetic relationship with similar viruses found in other regions, indicating its ability to adapt and infect diverse plant species. Recombination analysis identified 20 recombination events, suggesting a complex evolutionary history with major and minor parent strains. The metabolic profile revealed that infected plants had altered compound profile compared to the healthy ones, with references in gas chromatography, mass spectrometry, FTIR, and XRD analysis. These findings underscore the urgent need for effective crop protection measures and resistance strategies to mitigate the impact of Gemini viruses infections on the crop production. Thank you so much, all,
Well, thank you very much, Dr. Sana
for
Khalid.
hearing my presentation.
All right,
If
that
anyone
was
have
a
any
great
question,
share. That was
please
really a
ask.
great presentation from your side. And I would request the participants, if you have any queries or questions, you can ask Dr. Sana right now. Well, I think there is no questions from the participants. Thank you very much, Dr. Sana
Hello,
Khalid.
there.
Yes, let's talk. Yeah, Mr.
Yeah,
Tawfiq, please
it's not a
ask.
question. It's not a question though, but
Thank you so much. Thank you so much.
uh, Dr. Sana, I would like to tell you that I enjoyed your presentation, and I think that we have similar research interests, and I will be sending you an email right after this conference. I will try to collaborate with you and see how well we can work together. Thank you.
All right, thank you very much for your participating in our conference. Well, so that's an end. So this comes to an end of our virtual one-day program, and simultaneously we have successfully completed the live conference in Rome, Italy as well. So we have been fortunate to have a great identities from academics, industries, and other areas with us, and we'll be glad and thankful if you could join us in our next conference in person or virtually. The dates will be informed to you by email. Also, we request you to share your valuable opinion or feedback with us. For any further queries, you can mail to the concerned program manager, and certificates will be emailed to you. Thank you very much and have a nice day.